And so, so on that sheet, what was the trend with the surface area to volume ratio as the cell got bigger? So the cell got bigger. Colby.
The ratio went down.
The ratio should have went down. All right. Or sorry, when the surface area to volume ratio, when the cell is bigger, is a smaller number than when it is a small cell. So let's talk about why that is. So what does that mean when that ratio goes down? What does that mean in terms of, because it's a surface area to volume ratio, surface area to volume. So what does that mean with that? Okay, so the cell is maybe getting ready to divide when it's that big. Is it better, do you think, to have a larger or smaller surface area to volume ratio? It's actually larger. The larger the surface area to volume ratio. So if this number is, does that make sense?
I thought you meant larger surface area.
Oh yeah. So we want a larger number is best. That means that there's more surface area than volume. That's going to give you a larger number there with the numerator there. And so, why is that important? So volume is the amount of space that the cell takes up, so the literal space. And the surface area is the surface exposed to the environment. So why is it beneficial for a cell to have a lot more surface area compared to its volume, compared to the amount of space it takes up? Why is that a beneficial thing? Because it's a huge thing for cells to be able to do.
Because a lot of the stuff inside a cell requires nutrients and stuff from outside to come in and has to export other things. So by having a larger surface area, it's easier for stuff to go through within the cell membrane.
That's right, that's right. So if you have a large surface area, more things can go in and out, and then compared to the volume, if you have a smaller volume, those things that come into the cell can diffuse and get around the cell and get to where they need to be, versus if you have a large cell, you have a lot of space, but you don't have very much surface area. The surface area doesn't increase with the volume. And so while you still have surface area, it's not as great as the volume itself. And so therefore, if larger cells, yes, things can come into the cell, but it's going to take, there's so much space inside the cell that it's going to take a while for it to diffuse through and get through the cell and be able to be useful for the cell. So cells are smaller cells are much better in terms of that. And so let me use an analogy with a block of ice. So if you have a huge block of ice, and I put that takes up a certain space, a certain volume sitting on this table, and then you have a same block of ice, same space and same size and so on, but in this one, I took a jackhammer to it and broke it all up into smaller pieces. So it's still the same amount of ice, it's the same volume, but it's in smaller pieces. Which one is going to melt faster, that block or this block? This block. Why? You've increased the surface area by breaking it up. So there's more of the air that's going to actually touch, so therefore the heat is going to get to all the little pieces and so on. So you've increased that surface area, and so it'll melt a lot faster. Versus this big block over here has less surface area, so the outside is going to melt first, and then it's going to melt from the outside in, and therefore it's going to take longer. All right, whereas the smaller pieces will melt a lot faster. That's an analogy to the same idea with cells, smaller cells versus larger cells getting things into and out of the cell. So, so I think we wrote these two things down yesterday. And fix it. There we go. And so as a cell increases in size, its volume grows more than the surface area. So when a larger cell, it's going to have more volume than it does surface area, and the volume increases at a greater rate, and so therefore that's where you get that smaller surface area to volume ratio. And so this picture here shows what you just did. So here's a smaller cell and a larger cell, so it gives the surface area and how to figure that out. The sides are 1 unit, whatever that unit is, as a surface area of 6, the volume of 1, so surface area to volume ratio of 6. This is 5 units on each side, so here's its surface area. So notice here the surface area grew, but so did the volume. The volume grew as well, and the volume grew at a greater rate than the surface area, and so therefore when we do our ratio, it's a much smaller number. I like this picture because it shows what I was just talking about with the block of ice. So if these were cells, one big cell versus many smaller cells, the volume is still the same. So the volume, we have 1 unit on each side, and so you get the volume of 1 cell and multiply it by how many cells there are in here. And so the volume, or the amount of space these two take up, are the same, except this one is made up of many smaller cells. But the surface area is not the same because each cell has its own little surface area, and there's space in between here. So if we're talking about getting things to the center of the center of the space, there's molecules and air and so on that can move through there and get to the center, versus here it's just on the outside. So we have a greater surface area and therefore have a greater surface area to volume ratio, which is good for cells. All right. So that's one thing that you need to be able to recognize and calculate is a surface area to volume ratio. All right. Now the rest of the chapter, we're going to kind of look at specifics about cells. And so we're going to look at basically eukaryotic cells, and we're going to look at plant and animal cells and their organelles. So as I said yesterday, it's a kind of a review. In biology, you probably memorize some definitions about what the organelles do. Today we're going to talk about how they work together to form, to do certain things, to work together for a common function. So there's these internal membranes that are wrapped around these organelles, and which are, remember, little compartments. And so it helps to make cellular processes occur there. It minimizes competing interactions and increases the surface area where reactions can occur. So I'll show those illustrations as we go along and look at the different organelles. Oops, sorry. So these organelles compartmentalize or localize certain cellular processes and reactions so they occur where they're supposed to. And so it makes the cell way more efficient by having all these different organelles that do different functions. So remember that the cell membrane is a double layer of phospholipids. We're going to see that other membranes in the cell are also a double layer of phospholipids. And plant and animal cells are your basic eukaryotic cells. These guys are, remember, the eukaryotic cells have a nucleus and have membrane-bound organelles. So plant and animal cells have a lot of similarities in their organelles. They also have some differences. For instance, plant cells have a chloroplast versus animal cells do not. So these are this is a typical animal cell that you might see. These are what the organelles look like, and so on. We're going to talk about each organelle. You need to be able to know what the organelle does and be able to know what it looks like. All right, so this is a typical animal cell, but as I talked about yesterday, not all animal cells are round. Not all of them look exactly like this. They have different structures based upon their function. So yesterday I showed you like the neuron, the end of the neuron in the skeletal muscle cell and communicating with it. Those are two different animal cells that look nothing like this particular cell. And so inside of it, though, it has these different organelles. Some animal cells have the flagella, the tail, to help with it movement. In humans, that would be the human sperm cell. And In plant cells, it has the chloroplasts here. So this is a typical plant cell. Has a big central vacuole that we'll talk about, and so on, that's unique to the plant cell as well. So those are just typical plant and animal cells. This just shows you the variety. So here's an animal cell that looks nothing like that picture that we just looked at here. So this is lining the uterus. It has the membrane actually curves up and down around here. That increases surface area. We'll talk a little bit about that. This is yeast, which is a fungus. This is a typical plant cell and a unicellular eukaryote. All right, so not all cells look exactly like those pictures. They can be specialized. So let's look at the things inside of there. So we're going to start out with the nucleus and the ribosome first. So hopefully you remember that nucleus contains, notice here it says most of the DNA. That implies what? That there's DNA somewhere else in the cell. Does anybody know there's other organelles that have some DNA in it? Viruses have DNA, but viruses are not within the cell, all right? They're separate. So I'm talking about, so here you have a cell. Here's the nucleus. The nucleus contains most of the DNA. What we're saying is inside of the cell, there's something else that contains DNA.
It's not the ribosome.
It's not, no, the ribosome, people, everybody, every hour, so, well, first hour and now this hour, have said ribosomes because I think ribosomes is where proteins are made, where they use the instructions from the DNA, but DNA is not actually found there.
Mitochondria.
Yeah, the mitochondria. So I'm going to put here, another place is it's in the mitochondria. We'll talk a little bit about that. That makes the mitochondria really unique. We're going to talk about that there's a theory that the mitochondria was actually a free-living cell at one point. Because one of the things is because it has DNA, because it's unusual. And there's one other organelle that has DNA. Our cells don't have this organelle. Not yet.
Chloroplasts?
Chloroplasts, yeah. And then ribosomes, so the DNA is located in the nucleus. Ribosomes are outside the nucleus, and they use the information from the DNA, the instructions, to make proteins. So protein synthesis is what ribosomes do. So we're going to look at the nucleus first a little bit more in depth, and then the ribosomes a little bit more in depth. All right, so here's your nucleus. So contains, as I said before, most of the cell's genes because there's some in the mitochondria, some in the chloroplast. And usually, this is the organelle, if you look at the cell underneath the microscope, this is the organelle that you can usually see without a doubt. So even with our microscopes that aren't super, super strong. So the nucleus you'll see is a dark spot, large dark spot in the cell. It has, as I've talked about before, a membrane around it. That membrane is called the nuclear envelope. It encloses the nucleus, separating it from the cytoplasm, so keeping like the DNA and everything inside the nucleus from the cytoplasm. That is a double membrane. Each membrane consists of a lipid bilayer. So remember that's like the phospholipids, like the cell membrane lipid bilayer, like that. I'll show you a picture of this here. So, this is a cross-section of your nucleus. And so, what we just talked about the double layer. So this right here is enlarging that, so you can see that. And so here is the double layer, and these are pores that are openings to allow things in and out of the nucleus. So this double layer, when we just said that it's a lipid bilayer, so the double layer, I don't know if you can see this layer has a lipid bilayer, and this layer also has a lipid bilayer. All right, and that's what that means. That's what that is. And so, so that's the double membrane. And the pores let things in and out of the nucleus. What's inside the nucleus and stays in the nucleus is chromatin. Chromatin is if we look down here, is DNA wrapped around, so if this is DNA and the string is DNA, so this is DNA wrapped around these other guys here. I'll use a different color. These guys here are proteins. And so that's called chromatin. And so, so the DNA is wrapped around these proteins. DNA is very stringy. And so it's a very long molecule and very stringy. So if I use the analogy of a piece of sewing thread, so sewing thread here, if you have a large piece of sewing thread and you try to condense it and put it into a smaller area like the nucleus, it would, if you bunched it all up, it would get all tangled up and so on. And so what DNA, there's the same thing with DNA. So what we do with our sewing thread is we put it around a spool, right? And that organizes it so that it doesn't get tangled up and so on. That's kind of like what these proteins are like. So the proteins are a way to organize the DNA so it doesn't get all tangled up. And so that's what chromatin is. So it's not just DNA by itself, it's wrapped around these proteins. And so, so that's your chromatin. And then often you can see a dark spot, like when you look underneath the microscope, you can see the nucleus, and you can also often see a dark spot in the nucleus. That dark spot is called the nucleolus. That's where ribosomes are made. We're going to write that here in just a second. So ribosomes are made there. So that's the structure of the nucleus. I do want to point out one more thing with this picture, though, is that they added another organelle here, the rough endoplasmic reticulum. Why they put the rough endoplasmic reticulum on the same picture as the nucleus is because they're connected. If you see here, the membrane, the double membrane here is connected to the rough ER right here, and so therefore that membrane is continuous, and that's going to become important later, all right, with that. Okay, so let's talk about the parts that we just looked at. So let's write about it there. So the pores regulate what goes in and out of the nucleus. In the nucleus, DNA is organized into discrete units called chromosomes. I'm going to relate here in the next first four bullets here about the difference between several things: DNA, chromosomes, and chromatin. That's a lot of words that people get mixed up with. Each chromosome is composed of a single DNA molecule wrapped around proteins. And the DNA and proteins together are called chromatin. And then chromatin condenses to form chromosomes when a cell prepares to divide. So let me illustrate that here. So we're right up here before cell division. So the cell's just hanging out. It's not dividing. All right. So doing nothing special. When you look at it underneath the microscope, like I said, you can see the nucleus, but inside you can't see the DNA before cell division. So you know that there's some DNA in there, but you can't see it underneath the microscope. And let me explain why you can't see it. So the DNA, remember I said, is wrapped around these proteins. So I'm going to put in a red here. These are your proteins. It's going to make little balls here. And I'm going to say that these are proteins. They're actually called histones, is the name of them. And then in blue here is your DNA. So as I said, the DNA is wrapped around these proteins like sewing thread in the spool. All right, so this is DNA. And this is very loosely packed, meaning that so the DNA wraps around and then it wraps around again and so on. And so it's more still stringy, even though it still has these spools. It's very loosely packed, not really condensed together. And because DNA is so skinny and these proteins are so small, when they're loosely packed like that, you can't really see them that well underneath our microscope. But what happens is when, I should say that this is called chromatin. So when a cell's just hanging out, not going through cell division, it's in that chromatin stage, loosely packed, and you can't see the DNA. What happens then is during cell division, all right, so when the cell is dividing, what you can now see is, if this is the nucleus, you just can see dark spots in it. Now you can't see the individual pieces of DNA and so on, but you start to see dark spots in the nucleus. Now why can you see the DNA here and you can't see it here? It has to do with this loosely packed versus tightly packed. So what happens is this DNA wrapped around these proteins, very loosely packed, still kind of stringy. When it gets ready to do cell division, all of that kind of comes together and forms a short, stocky, thicker structure called a chromosome. All right, and so that becomes this, where the histones now are packed together, and the DNA is still wrapped around it, and it kind of condenses and becomes tightly packed. And that is what we call a chromosome. So really, we only have chromosomes. It's only in this format when the cell is getting ready to divide. Otherwise, it's in this format. And so we have 46 pieces of DNA. So in us, in our nucleus, there's 46 pieces of DNA wrapped around the proteins. And so then when our cells get ready to divide, all 46 pieces condense, and that's why we have 46 chromosomes. All right, and that's what happens during cell division. And so going back to this picture here, this one. So this is the chromatin there. So this is the loosely packed, and then this is it tightly packs together and condenses together will form ultimately the chromosome. All right, so that's the relationship between those. All right, so and then that dark spot is the nucleolus. And as I said, the nucleolus, basically its job is to help make ribosomes. So notice it says here is located within the nucleus and is the site of ribosomal RNA synthesis, or rRNA. We talked about before what kind of RNA? mRNA, right? And that was a messenger RNA. This is ribosomal RNA, and it's used to make the ribosome. So the ribosome is made out of ribosomal RNA. So when we say then this helps to make ribosomes. So the nucleus, I should say, so helps make ribosomes. That's the job of the nucleolus. All right, and that's the nucleus. So attached to the nucleus can be ribosomes, and ribosomes can be attached to things or not attached. So what are ribosomes? In biology, you probably just looked at the look like little dots, and they make proteins. They're actually a little bit more complex than just little dots. They're made up of RNA and protein, and they actually have two subunits. And they do carry out protein synthesis in one of two places. In the cytosol, we call those free ribosomes because they're floating around in the cytoplasm free, not attached to anything. Or they could be attached to stuff. So the first thing that they can be attached to is the nucleus. The second thing that they can be attached to is the endoplasmic reticulum. So both of those, they can attach, and we call ribosomes that are attached, we call them bound. They're bound to the membrane of either the rough ER or the nucleus. No matter where they're at though, they carry out protein synthesis. And so here's your ribosome. They look, like I said, they look like little dots. Really, they have a large and a small subunit made out of proteins and rRNA. All right, and that's where we're gonna make our proteins, and that's basically it for the ribosomes. Okay, so now building on the nucleus, we're gonna talk about. Remember, I said that there are several organelles that work together to perform a common function. Those organelles collectively make the endomembrane system. So the nuclear envelope on the outside of the nucleus, the endoplasmic reticulum. What's the relationship between those two? What did we just say? That they are connected, right? The membranes are connected. Golgi apparatus, lysosomes, vacuoles, and the plasma membrane. These guys, endomembrane. Endo means inside, so they're membranes inside the cell. And so what we're going to see is that they're either connected physically to each other or through what we call a transport vesicle. So these components are either continuous, like the nuclear envelope in the endoplasmic reticulum, or connected through vesicles. And I'll draw and explain what a vesicle is in just a second here. So let's look at the endoplasmic reticulum. Oftentimes this is shorthanded by ER, endoplasmic reticulum. And in notes and things like that, you can shorthand ER, but like if you're doing something where you're going to turn in or on an essay, you should write out the words endoplasmic reticulum. All right, so lots of them in cells, more than half of the membrane in a eukaryotic cell. As we saw in that previous picture, it's connected to the envelope around the nucleus. And there are two regions or types of ER called smooth and rough. The smooth ER lacks ribosomes. The word smooth is referring to the outside of it. The outside of it appears smooth because there's no ribosomes attached to it. The rough ER has ribosomes attached to it, so when you look at the surface, it appears bumpy or rough. Can you move this? So let's look at the difference. They're connected. They're both called ER, but they do very different things. So we'll look at the smooth ER first. And so just to show you, kind of looking similar to the nucleus picture here. Here's the nuclear envelope, so you can see here where it's connected to the rough ER, so there's some connections here. And then the rough ER is studded with those dots, those are the ribosomes. And then connected to the rough ER, you can see that right here, are these shorter, kind of more skinnier, I should say, ER, and that's the smooth ER. You don't see any ribosomes attached to it. So the rough ER is more like elongated sacs, all right, that are kind of flattened like this, and the rough ER, the smooth ER, is skinnier that way, more tube-like. And so that's how you can tell the difference between those. So let's look at the functions of the smooth ER. So synthesizes or makes lipids, breaks down or metabolizes carbohydrates, detoxifies drugs and poisons, and stores calcium ions. Calcium ions are very important for muscle contraction. So in skeletal muscles that make your skeleton move, there are a lot of specialized endoplasmic reticulums that store tons of calcium ions. Calcium ions are required to make your muscles contract. Let me talk a little bit about the detoxification of drugs and poisons. So one of the drugs could be alcohol. And so one thing I want to point out with smooth ER, all eukaryotic cells have smooth ER, but not all eukaryotic cells have the same amount of organelles. So depending upon what the purpose of the cell is, some cells have more of an organelle than other cells. So for instance, skeletal muscles need a lot of energy for your muscles to contract, to move your skeleton. So skeletal muscle cells have a lot of mitochondria compared to maybe a skin cell. The skin cell has mitochondria, but just not as much. And so same thing with the smooth ER. So all of this, the smooth ER, the liver, your organ in the liver does a lot of these things. That's one of the functions of your liver. And so therefore your liver cells have way more smooth ER than other cells of your body to help with these functions. And the thing is that also the number of organelles that you have is not always stay the same. Sometimes we can make more of an organelle. So going with alcohol, have you guys heard that if somebody drinks a lot of alcohol or maybe is an alcoholic, that the more you drink, you don't get the same effect and you have to drink more over time? Have you heard that? All right. This has to do with the smooth ER. So what happens is somebody who is putting a drug like alcohol into their body is trying to metabolize that and get it out of the bloodstream. That's the job of the liver, and your liver cells, the smooth ER in your liver cells, are working overtime to do that. So those liver cells begin making more smooth ER, so therefore you metabolize the alcohol faster, which means somebody that's trying to get a feeling from drinking alcohol needs to drink more of that, and so therefore to get the same feeling as before. And what that does is eventually destroys your liver cells, and therefore that's why alcoholics oftentimes have issues with the liver. All right. Question?
About like why it fails in
Yes, it eventually fails because it's making more smooth ER, but it also it can't metabolize it fast enough, even though it still makes it, and so it does destroy the cells. You know. And so that actually happened to my neighbor across the street who hasn't drank in a long time. He's a recovering alcoholic, and just this last spring he got a liver transplant. He hasn't drank for a long time, but his liver was, he was on a liver transplant list for years and years, and finally on the next this last spring he had a liver transplant, but he has to go in and you know, get tested periodically and go in and they test to see, they make sure that he's not doing, he's got a whole list of things that he can't eat or he can't have any alcohol. He said he can't even use mouthwash because mouthwash has alcohol in it, and they can detect that, you know, and then they'll put him on the list where he can't, they won't help him again, you know, so anyway, just an interesting thing here. And so that's what this last bullet was about, so that the liver cells have a lot of these, all right? All right, so that's the smooth ER. So the rough ER, much different. The rough ER has those bound ribosomes, that's why they call it rough, and it secretes glycoproteins. Glyco, whenever you see the word glyco attached to something, refers to carbohydrates. So a glycoprotein is a protein that has carbohydrates attached to it. A glycolipid would be a lipid that has carbohydrates attached to it. And it distributes transport vesicles. So secretory proteins surrounded by membranes. Let's talk about this. Secretory proteins, these are secreted from the cell. What does secreted mean? It leaves the cell, all right? So and it helps to make membranes. So I'm going to try and illustrate that here in a little drawing. So the board is the cell, so this is cytoplasm. This is the nucleus. I'm drawing the nucleus because we're talking about the rough ER, and remember the rough ER is attached, the membrane is attached to the nucleus. The nucleus has a double membrane, so I'm going to do the double membrane here. The rough ER is connected, so I'm going to show here a connection. Not the greatest artist here, so bear with me. So the rough ER, if you remember, looks like these flattened like little sacs here. I'm just going to draw one of them. There's many of them, and so on. And I'm going to put here RER for rough endoplasmic reticulum. So that's what it is. What am I missing from the rough endoplasmic reticulum? That's pretty important. The ribosome, right? So, so attached to it, I'm just going to put right here. This is your ribosome. Okay. So I'm going to try and illustrate these bullets here. So the ribosome makes what? Protein, right? Which are chains of amino acids. So the ribosome hooks one amino acid to another, and so on and so forth to make this polypeptide chain, which will then curve and so on and make a three-dimensional protein. So your amino acids are hooked together. So I'm going to just illustrate that with AA. All right, and so we get a polypeptide chain. Eventually, notice as it's building the amino acids, the amino acid chain goes into the rough ER. So eventually, when this ribosome builds its chain, that chain, I'm just going to draw it here as a straight line here. So this is your polypeptide. What happens next? It says, so oftentimes these proteins, it says secretes glycoproteins. Oftentimes with the polypeptide, they'll put little carbohydrate chains on it and so on. That's what that means. And so then next bullet says distributes transport vesicles. And so the transport vesicle, what happens is this polypeptide comes to the end here. So here's your polypeptide. What happens to the membrane of the ER is that it kind of buds out, it balloons out. So I'm going to draw that here. I'm going to erase that and show this coming out like this. And what happens is it pinches like this. But before it completely pinches off, this polypeptide, this chain, goes into that ballooning of the rough ER. And then this continues to pinch until eventually it actually pinches off. And now you have, this is called a transport vesicle. So it has a polypeptide that was made here in the rough ER, and it's transporting it somewhere, all right? So somewhere else in the cell. And so where does that go? And that's the next one, the Golgi apparatus. Question?
Okay, so when you do that little like smooth line polypeptide, is the AA like are all those polypeptides as well?
No, I was trying to show you to make the polypeptides, you hook amino acids together. So the ribosome is hooking amino acids together. Eventually then, when it's all done, you have this polypeptide chain.
Okay.
Okay? And so we have the transport vesicle. So where does that transport vesicle go? It's going to go to the Golgi apparatus. And so the Golgi apparatus. And so this consists of flattened membranous sacs. So it looks similar to the rough ER, except it doesn't have ribosomes. And its functions. It modifies the products of the ER. So the product of the ER was this polypeptide, so this transport vessel is going to go to the Golgi, and it's going to add some chemical groups, chemically change it a little bit. It actually can make certain macromolecules itself, so it doesn't have to modify things that are made in the ER. It can make things itself. And then it takes those two things and sorts and packages them into transport vesicles itself. And this is what the rough or the Golgi apparatus looks like. So I'm gonna talk about that as I'm gonna draw it and add to this picture right here. So this transport vesicle that just left the rough ER, that's this little guy right here. So this is the transport vesicle. Yeah.
I think you missed the last bullet.
Oh, did I?
Right, or maybe I did.
I
Okay.
didn't see it on the slide.
I don't have it on the slide.
Production of blank was the last bullet.
Might be on the next.
No, it's production of membrane. What's interesting is that first hour nobody said it. So what's interesting about that? Otherwise, I probably would have fixed it before this thing. It's funny. All right, so so that's the transport vesicle. So now what's happening is, notice here the transport vesicle fuses with the Golgi apparatus. I'm going to attempt to draw the Golgi apparatus here. So I'm just going to Remember, it's a bunch of these sacs here, and so I'll just draw a bunch of them like this, all right, and then like this, all right. And we have to label it because if we don't, we won't know what it is. And so that's your Golgi apparatus. So this transport vesicle from the rough ER comes over here and fuses with the Golgi apparatus. So that's that transport vesicle. Now that polypeptide enters into the Golgi apparatus. And the Golgi apparatus, like I said, chemically modifies it, puts some chemical groups on it, and so on, and just puts the finishing touches on the polypeptide chain, and helping it to get into its three-dimensional shape and so on. And this arrow here means that the polypeptide is going to move through the Golgi apparatus. So the polypeptide, that's what this is representing. So polypeptide moves through, and I'm just going to put GA for Golgi apparatus. And so in my drawing over here, here's the polypeptide. It's going to move through the various sacs, all right, and eventually end up over here. So then what happens is now this is modified and the protein is now finished and in its correct shape. What happens is the Golgi apparatus buds and makes its own transport vesicle. So it buds off here, and that finished polypeptide goes into that transport vesicle. That transport vesicle is going to pinch off eventually, and we have a transport vesicle with a finished protein here. Let's call it transport vesicle. So we wrote, let's go back to here real quick, back to the rough ER. Rough ER makes secretory proteins. So this protein that has now come off the rough ER and gone to the Golgi apparatus, it's supposed to be secreted from the cell. So let me illustrate that on here. And so on this, let's say, I still have this up from last hour here, this part right here. Let's say this is the cell membrane. So this is the cytoplasm, that's the nucleus, and so on. So the cell membrane of the cell. So secreted from the cell means that it's going to leave the cell. So guess what? This transport vesicle is going to come over here, and how is this protein going to leave the cell? It's also going to fuse with the membrane of the cell, and that polypeptide and that protein is secreted. So the protein is secreted from the cell. So one of the functions that we wrote of the rough ER and the Golgi apparatus is it helps to make membrane. Can you see that when this vesicle fuses, this vesicle has a membrane, when it fuses with the cell membrane, it's made more cell membrane? You see that? All right. And so therefore that's why it says that. And so it's secreted from the cell. Let me give an example of when a protein would need to be secreted from the cell. We've talked about our digestive system a little bit with digesting macromolecules. So let's say your stomach. Your stomach is an organ. It's hollow so that you can put, that's where your food goes, and the space inside. So if I were to just do a cross section here of your stomach, your food is in the stomach, so it's hollow in there, but the lining around the stomach is actually made up of cells. So we have cells here. And so what digests your food? Enzymes, right? Enzymes are proteins. So guess what? How do the enzymes get to mix with your food in your stomach? Where do those enzymes come from? They're made in the cells surrounding your stomach. So this could be a stomach cell. So your stomach cell, the rough ER in the ribosome makes the enzyme. It gets modified by the Golgi apparatus, and now this finished digestive enzyme fuses with the cell membrane, so that would be right here, and the protein is secreted. And for this case here, it's secreted into that space where your food is, and then the enzymes can work to digest your food. So therefore, it wouldn't be helpful if that enzyme, that protein, stayed within the cell, because it's an enzyme that works outside of the cell. So that's an example of that. And so that is, all of these things are what we call the endomembrane system. Why? Because they're either directly connected, like the nucleus, the membrane around the nucleus and the rough-ER, or connected via these transport vesicles. So what does that tell you about the cell membrane, the membrane of the Golgi apparatus, the transport vesicles, and the rough-ER, and the nuclear envelope? The membranes are very similar in construction, so that's why they can fuse together and so on.
What happens, like, okay, when you drink a protein shake, and those just make the proteins already made, then you just exit?
Okay, so when you drink, all right, that's a good question. So instead of writing food, you have protein from your protein shake, right? All right, proteins, you just drank proteins, which are chains of amino acids. So your food is a chain of amino acids. You cannot use this whole big, large chain of amino acids. It's too big. So when you digest the protein that you ate with your protein shake, these enzymes help to break the bonds between them, all right? And then you have individual amino acids. And then eventually you'll get to your small intestine, which will go into your bloodstream, and then your blood can take the amino acids all over the skull.
So, before I always wondered why not everything was just grouped towards the center to make it faster, it's probably there, but I'm assuming the reason the Golgi apparatus was separate so that it has more surface area instead of being directly against it.
Yes, absolutely. Very good. Absolutely. We're running out of time. So, we'll pick up this fun tomorrow. All right. I think I forgot to move this. So, you're watching the video. This is the picture.